In the previous videos, I explained how the disk is divided into different compartments that are molecularly differentiated, and in this video I'm going to talk about everything related to the growth and establishment of the shape of the disk. First of all, I'd like to talk about the concept of the compartments, which has already been introduced, but not enough. So compartments are defined because they are regions which cells do not intermix. So neither a cell nor its descendants ever move or migrate into another compartment. Here you have, for instance, this pink clone. So the cells in the pink clone divide, but never cross the boundary, never go to the posterior compartment, and the cells that are in the blue clone that is located in the posterior compartment never cross the boundary and never go into the anterior compartment. Here you have pictures showing the same. The black clone, which is in the ventral part, does not cross to the dorsal part, which is defined by the expression of apterus, and this other black clone, which is in the dorsal part, never crosses and never goes into the ventral part. The establishment of boundaries is mediated also by differential cell addition, but also by because there's a higher tension in border limits. So when cells of different fates are in touch, the borders have an increase in actin expression, and the tension of these borders increases. The mechanical tension in the borders is higher. So when you have a clone of one cell that has a different fate than the surrounding cells, this cell is extruded, it's eliminated from the tissue. If you have several cells, some kind of cyst is formed, and if you have a wider region, you get compartments. Okay, you get boundary compartments. So here is an experiment showing how this concept works. You have cells that clones that are overexpressing cubitus interruptus, that as you already know is expressed in the anterior compartment, because in the posterior compartment it is repressed by engrails. So cubitus interruptus helps to establish the anterior compartment identity. So clones in the anterior compartment have different shapes, and clones that are overexpressing CI in the posterior compartment are rounded, because they are minimizing the area touch, the contact area between the clone and the outside of the clone, and they have an increase in actin, which is in red. They have an increase in actin in the borders. Here you have like an scheme of what happens with the shape of the clones, and here you have a magnification of showing these are not CI clones, these are different clones, but the same concept. They have a higher actin expression in the border of the clone, so the tension is higher and the area of contact between the clone and the outside region is minimized. So then I'm going to talk about how the growth of the disk is controlled, mainly through these three mechanisms. The regulation of cell division, the orientation of the cell division, and also the regulation of changes in cell shape and size. This is an important concept to take into account. It's a Herwig rule, which states that cells divide along their long axis. Okay, so here in the top region you have a cell that is symmetrical. It's so it can divide. It can divide in any direction because it doesn't have a longer axis. However, in the lower region you have two cells which are not radially symmetrical, so they have a longer axis and a shorter axis, and their division will be determined by this. So they will divide perpendicular to the longer axis. Okay, so this is a rule that is followed, or has shown to be it's very common that cells follow this rule when they divide. Growth in the disk has been studied carefully. So there are many studies quantifying how much growth there is in each part of the wing. Here's the plot of one of the most complete studies, or one of the studies in which the amount of data is bigger. This data comes from studying many clones, so they induce hundreds of clones in different parts of the disk, and they study how much they grow, at which stage, and in which direction they grow. So in the top part you have the amount of growth at different stages. So as you can see, at an early growth phase, the center of the pouch, here's the pouch, which is the part that is going to give rise to the wing. The center of the pouch has a higher growth rate at the beginning, and then this overall the growth rate goes slower, but also becomes equal and homogeneous in all the pouch. Okay, as you can see here, the growth rate is the same in the center and in the periphery. So relative growth between center and periphery change. And in the lower part you can see the direction of growth. Okay, so in the center of the pouch, cells are growing mostly radially. So from the center, which will be the distal part of the wing, from the center to the periphery. Whereas in the periphery, they are growing in a tangential way, in a tangential direction to those cells in the center. And this direction of growth is maintained throughout the development of the disk. Many pathways converge to regulate growth, such as hedgehog, DPP, and wingless. Here you have vestigial, and most of these, the pathway that is most directly related to growth is the hippo pathway, which effector is Yorkie. Okay, and as you can see, vestigial regulates Yorkie directly, and most of these pathways in the end interact with the hippo pathway. One of the pathways which affecting growth is most well characterized is the DPP. Here you can see how DPP is required through all the wing disc development for growth. In this experiment, they knocked down DPP. They knocked out DPP at different stages during larval development. Here you can see the arrow marks the starting of the knockdown. So as you can see, if they repress DPP at the beginning, at the mid larval stage 2, the wing does not develop because the disc fails to grow. But if they knock down DPP later at the transition between larval and pup, there's still a slight decrease in size. Wings are like 91% of their final size. So at this stage, DPP is still required for growth of the disc. Most of the DPP effect on growth is mediated by repression of Brinker. As I already explained, Brinker is constitutively expressed in the wing disc, and then activation of the DPP pathway represses it. So this results in opposite gradients of Brinker and DPP. So here it is shown how mutation of Brinker and overexpression of DPP have the same effect on the disc. Okay, so in the top figure you can see the wild type. In red you can see PMAT, which is a readout of DPP signaling. Then in green there's a Brinker reporter, and in blue there's BRDU, which marks cell division. Okay, BRDU is a synthetic nucleotide which replaces thymidine in DNA when replication is happening. So when DPP is overexpressed, you can see how PMAT signal is spread all over the disc, and the Brinker reporter is repressed. Consequently, you have a growth of the disc, mostly in this anterior-posterior direction. Okay, and you have an increase in cell division, mostly in the margins of the disk, and maybe even a decrease in the center that later I will explain why is this. If you mutate brinker, so here you can see how your brinker signaling is very low, but contrary to the previous case, DPP signaling is normal, okay, completely normal. So you can see that PMAT signaling is normal, and yeah, you get mostly the same phenotypic effect, the same effect in size, and also the same effect in cell division. And if you use this double mutant, which does not have DPP signaling and does not have brinker signaling, you also get the same result. Okay, so in the absence of DPP, the disk is able to grow if there is no brinker. So DPP effect on growth goes through brinker. And here is more of the same. DPP hypomorph has a very small size of the disk doesn't grow. Then if you overexpress brinker, you get the same effect. And if you overexpress brinker and have constitutive signaling of DPP, which means that you overexpress basically both brinker and DPP independently, you get the same result again, a small d. So this DPP signaling cannot overcome brinker expression. Okay, besides controlling how much cells grow, it's important to control the direction of the growth. So as I already said, these divisions are oriented radially in the center of the disk. Here you can see, with this technique, which is called Revly, you can visualize many clones at the same time. So you can see when these clones are induced very early, like 24 hours after egg laying, and then you analyze the clones 86 hours after inducing them. You get big clones, of course, and these clones are oriented radially. Okay, as you can see, they go from the inner part of the disk towards the outside part of the disk. And when you induce clones later, you get, of course, smaller clones, but the pattern is the same. So this orientation of cell division is dependent on Dutch, which is an atypical myosin and constrains the borders of the cell. Okay, so Dutch is oriented, it's located in the most distal part of each cell, which the most distal is the part that is most in the center of the disk. Okay, so what Dutch does is to increase tension in this part of the cell. So when the cell is growing, it cannot grow in this direction because that is constraining it. So it grows in the opposite direction. It grows from the distal, from the center of the disk, which is distal, to the proximal part of the disk, which is the periphery. So the cell acquires this elongated shape, and because of the Herwig rule, when it is going to divide, the mitotic spindle is oriented with the longest axis of the cell, and the cell divides in this direction. So this results in elongated clones. As you can see, in a Dutch mutant, the clones have this random shape, whereas in a wild type, clones have this long shape. Okay, so that is required for this orientation of cell division. And then that's positioning specifically in the distal part of the cell, or this that's orientation depends on a gradient of forejoint and dashes. So forejoint is expressed mostly in the central part of the wing disc, and dashes is expressed mostly in the periphery of the disc. Okay, so there's like a reciprocus gradient. And as you can see, both of them are regulated by DPP. If you have DPP overexpressing clones, which have a constitutively active DPP receptor, so forejoint expression increases in these clones, and in these clones, dashes expression decreases. So here's more or less how the system works. Okay, so forejoin is a kinase in the Golgi reticulum, which and that shows and fat are two caderins which bind to each other. Okay, so what forejoin modifies the affinity of these caderins for each other. Forejoin decreases that shows affinity for fat and increases fat affinity for that shows. Then fat can modify this that is the myosin, which is responsible for the increase of tension in the cell border. So fat represses that location in the membrane and also other effects of that, such as growth. So when you have a clone that is overexpressing forejoin, okay, so in this clone that shows does not have affinity for fat. However, inside of the clone, fat have or has affinity for that shows. So fat goes to the border of the clone, okay, where it can bind to other dash shows that is outside of in the cells outside of the clone, which have affinity for fat. Okay, so fat gets here and binds to some dash shows that is outside of the clone. And since fat is repressing dash is not located anymore in these borders, these cell borders, okay, that are in overlapping clones. However, if you express dash shows, the opposite happens. So dash shows goes here to these borders that are in contact with the outside of the clone, and binds to fat. Okay, I mean that shows is in all the borders, and then fat is mostly outside of the clone, and that goes there where this dash shows is accumulated. So in the end, it's like a gradient of activities is created. So towards the center of the disk, you have this region that is rich in forejoin expression. So forejoin is repressing, is activating fat and is repressing dash shows. So the fat that is inside the cell goes towards contacts with cells that have more dash shows, which is cells that are in the periphery, because these cells in the periphery have more dash shows and less forejoin. Okay, so these cells with that have more dash shows bind to the fat in the region that is more central. Okay, or to the fat of the cells that are more central in the disk. And the same happens with the rest of the cells. Okay, so cells tend to orient their fat towards the exterior part, because the cells that are more exterior have more dash shows, and cells try to orient their dash shows towards the most central part, because that is the cell that have more active fat, because it has more forejoin. So in the end, this results in an orientation of DAT's location, because DAT is repressed by fat, so it will not locate in this external side of the cell, and that's what creates a gradient. Okay, so this is how cells can read their position, and that gets polarized. And as you can see here, expressing dash shows in an ectopic gradient. Okay, this would be the regular gradient of dash shows, and then that gets oriented in this way. Okay, the red lines, and clones grow in this way. So if you express overexpress dash shows in an orthogonal gradient, you get that clones get oriented in the opposite way, and that is oriented also orthogonally. Okay, so here clones are horizontal rather than vertical, and here you can see how this is a measurement of the spindle orientation. You can see how it's shifted in the red bars. It's shifted 90 degrees to the original division. Okay, and well, this results in the adult in a change in the shape of the wing. these wings that have an opposite gradient of dust jobs they grow less in the proximo-distal axis, but they grow more in the anterior-posterior axis. However, these clones that are oriented from distal to proximal are only the ones that are in the center of the disc. The clones like the ones in the figure B, okay? The clones that are in the periphery here are oriented in a tangential way, like around the circle, let's say, like in figure C. Okay, so how is this possible if the DPP gradient is the same, in the same direction? Okay, the Darcius gradient is in the same direction in the whole disc. So how is this possible? A hypothesis for explaining this difference in cell division orientation is that cells in the periphery are under different stress conditions than cells in the center. This means that since they are experiencing different forces, they are elongated in different directions, and because of Hurwitz's rule, they divide in a different direction. Okay, so in order to test for these different stresses in different cells, they perform laser ablation experiments in which they cut cell junctions and then they measure retraction of the surrounding tissue. So if they, as you can see in the second picture, these junctions, this surrounding tissue has retracted. Okay, it's like cutting some string. The string retracts very fast, and the more tension there is, the faster the string retracts. So this is what they measure. They measure the velocity of tissue retraction surrounding the junction that has been cut. So they perform laser ablations for junctions in different orientations, the ones that are lateral and the ones that are proximo-distal, which are the yellow ones. And they do this in the proximal part of the wing, which is the blue one, the periphery, and in the distal part, which is the center of the disc. And they find that in the proximal part, in the blue part, the difference in tension between the yellow junctions and the lateral junctions in red is very high. Okay, as you can see here, these proximo-distal junctions have a higher tension that is much higher than the lateral junctions, and in the center this difference is not that high. So they the reason is that since cells are dividing more actively in the center, these cells are pushing towards the outside. Okay, so cells in the outside, since the inner part is growing, cells in the outside are stretched in the circle gets bigger, so they are stretched, and this is why these yellow junctions are under higher tension. Okay, so this results in an elongation of the cell in this direction. So because of Hurwitz's rule, when the cell divides, it does it in this direction, which is different from the direction in which cells in the inner part divide. Additionally, as we mentioned before, although cells in the central part of the disc grow faster in the first stage, later the growing rate of the distal and proximal part of the disc become equal or more or less equal. Okay, so how is this possible if the DPP gradient DPP expression is still higher in the center? Okay, well, as cells in the center grow, they experience also a higher pressure. They are compressed because of their own growth, and after some threshold, this compression starts to repress their growth. Okay, so growth is repressed by compression. So if you overexpress DPP, as we said before, it causes overgrowth of the parts in the periphery because they are not under pressure, but it causes a reduction in growth in the central part, because these cells are already under much pressure. So if they try to grow more, they suffer even more pressure, so they just cannot grow. Okay, their growth is repressed. Actually, the hippo pathway is repressed by this pressure. So here you would have a model of the pressure, how pressure affects cells. So in the outside, cells are stretched, so they are stretched and they elongate in this direction, resulting in cell division in this direction. Also, because stretching and elongation increases division rate, and cells in the center are compressed because of their own growth, so their growth rate decreases. Finally, it's important to mention that the Dashaws and four-join system also regulates growth. Okay, so that's myosin represses Ward's kinase, which in turn represses Yorkie. Okay, so whenever that is present, Yorkie is active and can go into the nucleus and activate growth. If that is not present, maybe because there is not a gradient of Dashaws and four-join, if this gradient is absent, then since fat is all over in all the cell junctions, that is inactive, and Yorkie gets repressed, gets phosphorylated by Ward's kinase and cannot go into the nucleus. Okay. However, if you have a fat mutant or that's just mutant, you have a lot of that. And therefore the Ward's kinase is completely repressed, and you have too much Yorkie, which induces a lot of growth. Indeed, you can see as both DATs and fat mutants have a growth that is not directional compared to wild type, where clones are elongated. So both in both DATs and fat mutants, clones are more or less round or amorphous. Okay, but DATs mutants are smaller, clones are smaller because they grow less than wild type, whereas fat clones grow much more than wild type clones. Okay, this is because of this experiment. And now, how do all these mechanisms influence variation in shape in the adult wing? Okay, so basically a failure to orient the growth radially, as we said, will affect the roundness of the wing, and we already showed pictures of that when this Dashaws gradient was reversed and was induced in an orthogonal direction to relative to the normal gradient, and you saw how the height of the wing increased, whereas the length of the wing decreased. Okay, so changes in morphogen production or their different parameters, degradation, diffusion, or changes in the interactions between the morphogens can affect the direction of these four-join and Dashaws gradients, and therefore will affect the relative growth in the different axis directions. Okay. Additionally, changes in the fat and DPP or vestigial systems can lead in changes in the differential growth between the proximal and distal regions, which will also result in changes in the shape of the adult. And finally, as you have seen, apoptosis and cell intercalation processes do not seem to be very relevant at this stage in determining the adult shape of the wing. Another important topic in the development of the wing disc is the formation of folds. So there are three folds in the disc: the one between the hinge and the notum, the second between in the inner part of the hinge, and the third between the hinge and the pouch, which is the part which will give rise to the blade. Here there's a video of how these folds form. In order to study how these faults form, we can define first some morphological parameters such as apical length or basal length and apical indentation, which could be this. Okay, so in this middle row you can see data related to the middle fold, which is the hinge fold, and in the lower region you can see data related to the hinge pouch fold. As you can see in the middle column, both folds form because an increase in apical indentation. So as you can see in the red bars, apical indentation increases during folding in both folds. However, changes in cell shape are different between both folds. You can see how in this hinge fold, the basal length increases in blue, whereas in the hinge pouch fold, the basal length remains constant. Okay, and you can appreciate this in the pictures. Here you can see the basal length before folding and after folding, where cells are wider in their bases, and this does not happen in the hinge pouch region. Okay, so focusing first in the hinge fold, we can wonder why this happens, why these changes in the cell basal length take place. So they perform again laser ablation experiments, and they measure base tension in the basal region, and they see that cells are under much more tension in the basal part than in the apical part. So the cell junction are under more tension in the basal part. Also, they find that this tension is released if cells are treated with collagenase. So collagenase degrades the extracellular matrix. So they find that after treatment with collagenase, basal tension returns to values similar to those in the apical region. Additionally, when this collagenase treatment is performed, the basal area of cells increases. Okay, so a release in tension results in cells relaxing and having a wider area. So this is a reporter of Viking, which is one of the subunits of collagen 4, and you can see how this Viking is degraded, or its location decreases in the place where the hinge fold is going to happen. Okay, so you can see this is before the folding and this is after the folding, and you can see how this Viking GFP decreases in this part. However, this does not happen in the other fold, in the hinge pouch region. Also, an ectopic expression of a protease in a stripe that is completely independent to the original place of the folding results in a basal decrease in tension. As you can see in this graph, tension decreases, basal tension decreases when you express a protease, and an ectopic folding results. However, as we already said, this basal increase of area does not happen in the hinge pouch fold. So what happens is an increase in actin, as you can see here, in the lateral parts of the cell. You can see here an increase in actin, okay, and also here. And this increase in actin, which you can see here in the purple line, is correlated with a decrease in cell height, which you can see as the dotted line. Okay. So what they propose is that this actin increases tension in the lateral parts of the cells. Therefore, cells are compressed, and this is how the hinge pouch fold takes place. These two folds are formed because of different mechanisms. For the hinge fold, cells are under big tension in their basal part, And when the basal matrix is the extracellular matrix of the basal part is degraded, this tension is released in the cells, and so cells relax and their basal area expands, and this causes the tissue to bend. In the hinge pouch fold, however, there's not degradation of the basal extracellular matrix. Instead, there's an increase in actin in the lateral parts of the cells, and then this increases tension and causes the tissue to fall. Well, these are simulations that show, these are 3D vertex models that show how these two mechanisms are enough to produce bending in silico. In the middle part, you have how a decrease in basal tension causes an expansion, a basal expansion of cells, and this causes folding. Then in the last column, you have an increase in lateral tension, which also causes folding. Okay, and in the first column, you have a mechanism that could be that had been postulated before, but in this case, it is not happening in the wing disc, and also in silico it is not enough to produce folding, and this mechanism is just the increase the increase of apical constriction. Okay, so this cannot form a fold in silico, and it does not happen in Drosophila. Finally, we what is regulating the place of the folding, okay, of this degradation? Well, there's wingless expression is basically it's in the dorsoventral boundary, and also in the boundary between the pouch and the hinge part. Okay, so in the hinge fold is coincident where the place where there's no wingless signaling anymore. Okay, and actually, if you overexpress wingless in this hinge part, in the place where the fold is going to take place, wingless can repress the fold formation. If you here, there's again an overexpression of wingless in a direction that is orthogonal to its usual direction. Okay, this red stripe here. So the folding is prevented in this part, and you can see that there's an increase in integrins here where wingless is being expressed in comparison to this place, which is the place where the fold is actually forming. Okay, so there's an increase in integrins. So integrins bind to the cytoskeleton and can provide an active tension in the cells, and also can mediate interactions with the extracellular matrix.